So I'm just going to give you an idea about what you can do with what kind of experiments that you can do in a clinical setting and how we have actually used this in our work, right? So I've titled my talk, How You Can Use Transcriptomics in Clinical Research. So I mean, right now I don't think we need to talk about this. So we are basically going to be concentrating on just this part, the transcriptomics. And so I mean, again, I think you're all now well versed with what is transcriptome, right? So I'm just going to skip this. But the only thing to remember here is that a transcriptome is a temporary state, right? It's a snapshot at one given time point. So it is not going to represent the cell at all the stages, right? So but in a bulk thing, you will have cells at different cycles, I mean at different stages of the development, but they'll all be fixed at one time, right? So it's a snapshot of that time. So that is one thing that you have to remember. And of course, there are different kinds of RNA, and depending upon the work that you design, you can have different libraries, and you can actually quantify all these kinds as per your study protocols. So the only reason that I've put up this timeline is to just show you that from the invention of the PCR in '84, it took actually a decade to come up with the real-time PCR. I mean, that is how long it took. And now in 2016, spatial transcriptomics was in the picture. So after that, development was really fast after the advent of next generation sequencing. So there are different kinds. My talk is basically about this. And tomorrow we will talk about the single cell RNA sequencing. And probably if we have time, they can tell us a bit about the spatial transcriptomics as well, which is probably of more interest to the pathologist, right? So different kinds of RNA sequencing platforms are available, and I'll be talking more about what we have used, which is the standard short-read RNA-seq as well as the small RNAs. And before I tell you all that you can do, I'll tell you a bit about the different repositories. So this is important because the moment you generate some data, it has to be shared. So there are different repositories that are available where you have to upload your all the data that you generate. So like commonly we use the gene expression omnibus. So you have to upload your data. It is checked by the curators there, and an accession number is provided. And only then you can actually publish your data, and you can decide when you would want this data to be made available to the public, and other people can actually download it and use it for their own analysis as well. So these are some other resources that are available, and you can probably explore them. So yeah, I mean by now you all know all this really well. So these are the different steps, different workflow in RNA-seq, and Francesco is kind of taking you through this, and he'll be talking about the next steps today and tomorrow as well. So I'm not going to talk about this, but what I do want to show you is a very simple experimental design. What we did, that's how we actually started, this work. So we had a disease condition and a healthy condition, and we really wanted to see what's the difference. Very simple question. I'm not going to talk about all the different factors that weighed in, but this was the basic idea. And again, after doing the simple steps of collecting the sample, which is actually not simple, there are many nuances to it, but I'll skip that. We can probably discuss that in some other day. I mean even, so even actually what kind of kits that you would use, what is the order of sample draw? That is also really important if you have to go for an RNA sequencing experiment. All these things will matter. So these are the certain things which you have to keep in mind before you design your experiment. And then you have your data which comes out. You have these different steps, and probably we are now at the HiSat2 stage. We are aligning our data to the reference, and we'll be using feature counts and dseq2. This we'll do today. So again, once you start doing your own work, this is just a brief about how it will look on your systems, right? So this is a snapshot when we are running the FastQC, the Trimomatic analysis, and HiSat2. So you're already doing this, so that's why I'm kind of not going into the details. So eventually you will have your dseq2, which you will be, now after this step you have your differentially expressed genes. So now, if you remember what Dr. Boni did yesterday, he took you through a very simple way to plot your data in R, and he actually arrived at this very beautiful plot, right? So there was a reason why he took you from that. So now I think most of us can do this. You will have to do this here because the data is huge. It's not a couple of hundred reads that you will get. It will be in thousands and lakhs, right? So you cannot manipulate it with the normal Excel or something like that. You have to do it in R. So once you have your data, now by using, remember, so we had put some, we had used the p-values and we had decided that I want to see the data or the DEGs which are above a certain cutoff or below a certain cutoff. So that is precisely what I've done here. The most highly up-regulated and down-regulated genes are highlighted here, and you can also have the names show up. So all of this can be done pretty easily because now you have the background, right? So once you have done this, you can actually, after this comes your visualizations. So how do you see your data? That's some part of it I think Dr. Boni will take tomorrow, but I'll show you what we did for a very simple experimental design. So I feel that, you know, transcriptomics can be used in multiple ways. So some of this, which are I think specific for our unique niche, would be disease-specific biomarker discovery. I think a lot of people in PGI are working on this. Disease diagnosis and monitoring. I'll show you some examples where we've done this. And understanding the disease mechanism. So precision medicine is something I have no experience in, but probably our other departments might be using this as well. And obviously, finally, for guideline development standardization. But that's a far way to go for us at least. So I'll just now give you one example where we've used transcriptomics to look at the disease pathogenesis. So our unit is allergy immunology unit. We work in the area of primary immunodeficiencies. Now they are called as inborn errors of immunity. So most of these have a monogenic cause ascribed to them. But a disease like CVID, common variable immunodeficiency, the genetic, there is no one monogenic cause which has been described. So in this case, to understand why a patient presents with a deranged immune profile is interesting. So for this, we've been looking at this disease in different angles, but I'm just going to talk about the transcriptomic part today. So very simple experiment design. We have a group of patients, we have healthy controls. So we just simply wanted to see what's happening in the neutrophils or in the innate cells as per your design. So again, simply collect your samples, do your RNA-seq, get the data, and then comes a visualization.
Is it PBMC?
So this is both PBMCs and neutrophils. So I'm just giving you just a snapshot of how your data would look like. It depends on your experimental design. What is your research question? So here in this case, so again, I could see that there are a lot of genes which are significantly upregulated and downregulated. So it depends on what is your criteria. How stringent do you want it to be, or how relaxed do you want it to be? So those are the parameters you can play with and see if it makes sense for you. Again, these are very good looking plots, good for the reviewers, but you need to understand what will you do with it. There's a huge amount of information that is available now. So again, you will have an idea about the genes which are upregulated and downregulated in a certain condition and the common ones. So from there, you can start building up your story. So you can see the function of these genes. For this, we have some different tools, but you can even start searching yourself. If you understand your disease, so again, you will have to revisit the literature, understand what is the pathogenesis, and then correlate with the data that you generate. So again, once you have your differential counts, right, expression counts, so you can actually go ahead and start making these plots. So this is called the gene ontology enrichment analysis. So what does this mean? So this picture actually gives you a lot of information. So what you can see is you can actually look at the different processes, where this is localized the cellular compartment, the biological process, and the molecular function as well. So the color of the bubble denotes the process type. The intensity is how significantly it is either up or down regulated, depends on how you plotted it, right? And finally, this bigger the bubble, the more are the number of genes in that particular geo process, right? So if it is a bigger bubble, so more genes. So by this, in one graph, you can have all these processes which you can actually see. Like for example, for me in this disease, I'm more interested in the immune responses. So can you see here? So regulation of immune response. Some of these hits are pretty highly significant, around 40 gene sets are present here. So now you have an idea, okay, these are the processes which are important. Then you can move ahead and there are many other visualizations. I'm just giving you a very brief overview. So you can do your path view analysis. In this, you can actually superimpose the genes which are up or down regulated into, these are all different pathways. Like for example, I chose this P53 signaling pathway. So you can see some which are in red. You can decide, right? For me, I've taken red to be upregulated, green to be down. It's up to you. So now you can very nicely see the data that you get, actually what is happening. So in a particular pathway or in a particular, for example, the B cell receptor signaling pathway, what is happening here? So I mean, for us working in a clinical setup, this would make sense, right? I mean, when we see this, we can actually interpret and correlate, like, you know, this is my patient and eventually this is happening. So now I know that in CVID patient, something is messed up with the B cell receptor signaling pathway. Actually, you will see a lot of hits here just for today's presentation, just not to make it too colorful. I mean, not to overwhelm everyone. So I've just kept these 1 or 2 genes just to show you. You will have this kind of analysis that you can actually do with your transcriptome data. So basically for us, you know, we got a lot of insights from this data. We saw that there was some immune dysregulation happening in our patients. We could find out that apoptosis and cell psychoregulation was impaired. So, you know, these are the kind of insights which you can get and find you can take this work forward.
These were made on p-value or adjusted p-value?
Adjusted p-value. I mean, again, we'll just see it in the examples today, and some of these visualizations Dr. Boni will take tomorrow. So you can just, I can show you some tools also that you can do online. There are some online tools also available. Just put your gene set list and it will show you these pathways. So we can go through that as well. So, a bit about how you can go for disease-specific biomarker discovery. This is another work that we did in Kawasaki disease. So this is now the commonest cause of acquired childhood heart disease in India. And in this, you know, we wanted to see if we can use our data sets and the publicly available data sets and see if we can find some potential biomarkers which we can further evaluate in a larger cohort. So for this, we had downloaded data, and then, you know, we had our own data. So we tried to see any commonality between the genes. So we found a couple of good hits. These genes which were found to be involved in neutrophil degranulation, they were you know, highly expressed in patients with Kawasaki disease. So this is something that we are actually following up in our lab. So again, this is how you can even actually use publicly available database, and you can, you know, take your own data, see if you find some commonality or some differences, and you can design some kind of a study. I feel this is something which we really do a lot in PGI. So, you know, rather than going the old, I mean, not the old fashioned, the regular way by going through the proteome, you can even start here. That is also fine, but that's another way to look at things, right? If you have a very volatile disease condition, something like that. So a bit about disease diagnosis and monitoring. This was something have you heard about this? How many read about this? Yeah, I think most of us, right? This was really, you know, something happening up, so many headlines, oh, black fungus, people are dying. You know, this was during the 2021 Delta wave of the COVID. We got a lot of upsurgence of mucormycosis cases, especially in India, along with aspergillosis. So we wanted to see that what is happening. So amongst the many different things that we were evaluating, we also kind of thought about looking at how the immune cells are responding. So what is different in these patients as compared to patients who present with only COVID, the healthy patients who we assume who were negative for the COVID PCR. So that was one group. One group is the COVID people who were positive for the COVID PCR. One group who presented with just mucor infection and no COVID, and the other group was the COVID and mucor, right? So this is a lot of factors here. We'll also talk about that later. So that study, and we were looking at the different compartments, the innate compartment and the adaptive compartment as well. So what we did was, I've just told you the methodology, and again we did all those analyses, and we finally found some pathways which were really upregulated in these patients, especially netosis. This is something, we would assume that this is something that you would see in pathology, but the moment you fix your tissue, the nets get degraded. So you will not actually see them. So here in the case, you can see all the genes which are involved in neutrophil degranulation, which eventually leads to netosis, were highly upregulated. If you can see, most of these are red, right? So that means in these patients, this was really something which was happening a lot. And it actually matches with the clinical phenotype that we were observing in these patients. So I'll not talk about this. And I mean, of course, by this you can actually study the disease mechanism. And this is about, so we also wanted to look at how, what is the disease progression? How does COVID-19 lead to mucormycosis, or does it there is a predisposition in these patients, or what is it? So this is some kind of analysis we are still in the process of doing. We've not really come to a conclusion, but the reason I show you this is that if you have like a disease progression, something like this can also be done, you know. You have at N0 stage, N1, N2, you can have different snapshots, and you can actually compare and see how your disease or process is evolving. Uh, Finally, I'll just one or two slides more, and we can go to the hands-on. So in this case, we also have a bit of a microbiology, because that's what I've trained in, can't let it go. So we've also looked at the biofilm. So there's a couple of papers which we published here, and we wanted to see the contribution of alternative transcript isoforms in the biofilms. So this is one work from where this entire story started. So we actually did not stop at just the gene ontology. We went ahead and looked at the isoform also, because this would really be important, especially in eukaryotes, right? Because we have alternative splicing. So different isoforms are generated. So what is the isoform usage? So if you just see, I'll just take 2 minutes. You can look at the gene expression in the healthy versus the disease condition. Then you will have the isoform expression. What do I mean by this? These are the different isoforms of one gene, and in the diseased versus a healthy condition, you can see there is some difference, right? And then you can actually see which isoform is being used at that point. So this is called as the isoform usage. So out of all the different isoforms, this is the only significant one. So, but then we did not stop here, we took it a step further, and we wanted to see how these isoforms will contribute to the different domain architecture. So this is an example of nuclear factor kappa beta 1. So now I can see that my complete protein would have all these domains. So this is the one protein which is, this is a transcript which is actually encoding for all these proteins. But is this the one which is highly upregulated or expressed, or the one which is used? And this is how you basically can actually see if the isoform which is significantly being expressed in your condition, is it the one which is forming these different proteins, right? So again, this is something which can give you a lot of clues about, is it, if you're not sure about the disease pathogenesis, this can be a major factor, especially in infectious diseases, that, you know, the pathogen can subvert the human response and, you know, change the isoform usage, make the protein which is convenient to the pathogen. So that is an upcoming mechanism which is being discussed a lot. So I'll skip with this and finally leave you with this thought, would you like to seek or not? So thank you, everyone. Mistakes.
Yes. Okay, good. My best friend. Good morning. I'm still a bit sleepy, I think. So if I talk too slow or you see I'm falling asleep, let me know. Just scream. Well, the goals for today, right? So today you're going to be working in R, finally, I guess. So the main goal today is going to be we're going to do a differential gene expression analysis using this R package called DSeq2. Again, we were going to do it hands-on. That's the main idea. But before that, I'm going to review some of the concepts related to DSeq2. This is what we saw yesterday, right? So yesterday, what we did was we went from the QCM filtering of our FASTQ files, we aligned it to the genome, and we used a string type plus that small tool to create our count table, our raw count table, or the count matrix, we can call it too. So today we're going to focus on differential gene expression analysis, and again, depending on the time we have, I don't know if it's going to be today or maybe tomorrow, we're going to use it. We're going to do pathway analysis maybe with this data or with the single cell data that we're going to analyze tomorrow. This is how we finished yesterday. This is our count table, you remember? Here you go, our raw count table matrix. The samples we did yesterday were just a test. Today, what I did is I got the actual full set of samples, and I processed them. And in your folders, when we log into our Jupyter app, you're going to have a full count matrix. So for differential gene expression, we have different options. I think I already mentioned that yesterday. We're going to use DSeq2. I would say that's probably one of the most popular ones, if not the most, with the goal of producing some tables with the differential gene expression analysis, log2 for changes, p-values, q-values. How many of you have seen one of those final tables with the gene counts, p-values, 1, 2? Oh, okay, more. So you're a bit familiar with it. Okay, so DSeq2, it's part of I'm going to just show it. It's part of the bioconductor. Well, how many people have heard about bioconductor or use R with bioconductor? A couple, okay, a little bit less. So in bioconductor, it's like a database. It's a repository of R packages, all related to bioinformatics. And DSeq is part of it. So bioconductor is well documented. There's a lot of information there. So I recommend you, if you're going to do analysis, you can always consult what is available there for you. It's all related to R, though. The advantage of DSeq2, so it allows you to create from very simple models to analyze your data for more complex models like, oh, I want to remove batch effects, or the example that I gave you yesterday that included not only one source of variability in your data, the condition, but there were other sources like genotype or I don't know what else it was, the sex of the animal, right? All those kind of things. You can create relatively complex models and still do an analysis with your data. It's not just, oh, I'm comparing wild type versus control. You can go farther than that. Well, this is just the paper, the original paper for DSeq2. Okay. To start the analysis with DSeq2, you need 3 pieces of information. As I said, your count matrix. You're going to need what we call the metadata. We talked about this yesterday too. So the information that you have about of the sample, right? Generally, is going to be the simple example is going to be you're going to have a few samples that are your control, and a few samples that you treated, or there's something special about it that you want to compare with it. These metadata can be more complex than this. Again, going back to the example of the mice being in cages and the sex and the genotype, you can create bigger metadata files. And you need your You need a pointer. That's where you need a better. And you need your design, right? So in this example, we'll be pretty simple. We are expecting that the condition that we are studying is the main responsible for the gene expression differences. And this is how we're going to express it in DSeq2. And I've been showing examples of this, right? You recognize that tilde here and how you express the formula after it. It's the same way that the linear models in R express the formulas, like for linear regression or anything else you do in those simple linear or generalized linear models. Here is right. So if you would do a linear regression, it would look like this, right? So in the formula, we skip the intercept or the baseline to just give the other parameters. But as I was mentioning, you can even build more complex models. And if we have time, we'll try to do an example with this. In the practical and the hands-on practical, we're going to start with the simple one. But if we have time, we'll go something as complex as this, if we can. OK, so if you are in R, the only two commands that you need to do your analysis initially just would be these two, nothing else. So in one, you're going to provide the matrix, the metadata, your design formula, and the DSeq2 call will do the analysis for you. And that's it. Pretty simple, right? Well, we're going to spend 2 hours just exploring the data probably and doing other plots and doing other things. But pretty much an analysis will consist in 2 lines. You can add a couple more to get the results, and that's pretty much it. So as I said, the count data, metadata, and your design. So when you run this, you're going to get some report that DSeq2 is doing some analysis. So we're going to talk about the normalization. The way that DSeq2 normalize the data, they call it, they use size factors. That's what we DSeq2 call them. And I'll show you how you obtain them. I'm going to give some details about how you do that. Again, it's one of those things that you're going to see once, and then you're going to forget about it. You don't have to memorize how all the steps of the analysis are. But yeah, we need to do normalization. And the normalization in DSeq2 is by scaling the counts with a size factor, meaning we're going to divide every count that we have in our data metrics with a size factor per sample. It estimates the dispersion of the data, meaning how much noise you have and how much signal comes from the replicates. And then we're going to use the generalized linear models to calculate the fit to the data and calculate the fold changes. Or if I say coefficient sometimes, I mean fold changes too. They are interchange. So that would be our slope in our regression. For in this case, it's going to be our fold change. And then, of course, it's going to estimate our p-value. Basically, it's going to test our hypothesis, the null hypothesis that is our gene being differentially expressed or not? Is the fold change different from 0 or not? OK, why we need normalization, right? And we talk about this a little bit too. And you guys already mentioned, OK, yes, every sample is going to be sequenced at different depth. So we need to compensate for that, right? So if you have a sample that is deeper sequenced, that sample is going to get more reads. That's fairly logic, right? There are other things we can consider for normalization. One is the gene length, and the other one is the RNA composition. Sorry. Right, here I'm giving an example, right? So we have sample A and sample B. You can imagine that if you sequence 10 times more sample A, you're going to get more reads for sample A. That doesn't mean that it's differentially expressed, right? It's just you sequence more. The other thing is for a short gene, you're going to get less read than for a longer one. That is going to accumulate more reads across the gene length. And I'm going to talk about RNA composition later because it's the harder one to, let's say, understand. But I'm assuming you understand the basics of gene length and sequence length, right? Why we want to, or why we would want to normalize for them. Yep. So here I'm listing some of the common methods or methods and units of normalization. You probably have heard, or I don't know, I definitely, when I started doing transcriptomics, this is where the most common ones are PKMs and FPKMs. You still heard them, right? We use them less and less now because we see problems with them. But this is probably one of the most common ones we've had for the past 20 years. I said that for DSeq2 we use the size factor, but if you use another program like HR, it's going to use a different unit. If you notice, most of them already take into account the sequencing length, right? They use some sort of normalization like dividing by the total number of reads or using sort of the division by the total number of reads. So RPKNs, FPKNs, and TPMs, they use the sequencing length, or all of them use the sequencing depth, but these two also use the gene length. Why do you think maybe it's not that important to use the gene length of a gene for normalization? Or why DSeq2, for example, that is one of the most popular tools that doesn't use the length of a gene for normalizing? Any ideas? I'm asking why it's not important, or why it's not so important that DSeq2 decide not to use the length of a gene for normalizing. Well, basically you are comparing one gene across samples. The length of the gene doesn't change. It doesn't matter if it's sample A, sample B, or sample C. The gene length is the same, so you don't need to normalize for that, in theory, right? An exception would be if you want to compare within a sample across genes, but we don't do that, right? What we do is to compare the same gene across different samples, and the length of the gene doesn't change. So that is why that more modern approaches, let's say modern approaches, they don't use the gene length for normalization. They use another thing, which is the RNA composition. Well, but this is another example, right? So here I'm giving what is important to normalize for the sequencing length. We had sample 1 that is bit sequenced to this depth. If you double every single gene, you get the double of the sequencing depth. But it doesn't mean sample 2 it's 2 times more expressed than sample 1, right? It's just been 2 times more sequenced. Okay, so now I'm going to talk about the RNA composition, or what we also call library composition. So there can be situations where you compare, for example, heart versus lung, or 2 different tissues. And you may find that only some genes are expressed in heart, and they are 0 expressed in the other sample, right? So this is a type of what we call different RNA composition, right? You already know there are going to be a bunch of genes that are expressed in one and completely turned down in a different sample. Another one could be if you have a KO, right? So if you knocked out the transcription factor, all of a sudden everything that transcription factor controls is not going to be expressed in your KO compared with the control. I mean, you want those situations, right? You want to know what genes are differentially expressed, but that creates a problem. So in this case, I put a really simple example here, right, with a few genes. So I know it's hard to imagine that it is a situation like this, but let's imagine you have this sample. Both of them are exactly sequenced with the same number of sequences, right? But all of a sudden, you knock out one of the genes, or for whatever reason in this sample, the gene is not expressed. So you're going to have to distribute these 627 reads across all the other genes, right? Because both of them are sequenced at the same sequencing depth. So what's going to happen is you're going to have to distribute them, but actually these genes are not differentially expressed. If you try to do an analysis where the RNA composition is not taken into account, you're going to say, yes, this sample 2 for this gene has 4 times the expression level is higher, right? So the rest of the genes are going to look differentially expressed. Actually, if you do an analysis with such a small table where the RNA composition is not taken into account, all of them are being differentially expressed, which is not true. The only one that is differentially expressed is the one that is knocked out. Yep, so that's what I'm trying to express with this simple example. What is the problem of RNA composition? When you have a bunch of genes that are knocked out, and you don't consider the problem of RNA composition in normalization. Okay, so this is the part where I'm going to go into a little bit into details. Again, you don't have to know exactly how it gets all the steps in the process. This is how DSeq2 normalizes, and I want to go through this a little bit just to show you how the RNA composition is taken into account in the normalization process. Basically, the first step is what is called getting the geometric mean for every row in your table. So basically, you just transform the every count in the natural log and then getting the mean. So that's the geometric mean, right? The advantage of using logs and everything else when you normalize is that you are removing the extreme cases, right? And focusing only on those genes that are, let's say, more normal express, right? The housekeeping genes and things like that. Another problem of doing the log transformation is, and in R, right, a 0, the log natural of 0, is going to be transformed to a minus infinitive, right? So DSeq2, what it does is removing genes that have a 0 count. So those genes are not taken into account when you do the normalization process. So meaning you are removing the RNA composition problem of genes that are only expressed in one sample and not in the others. Then you subtract the geometric mean to the counts, and it goes through this process. Again, you don't have to memorize all this. Then it goes for every sample. It calculates the mean of the ratios that you did. Now it goes back to denormalize. So it goes back to the natural number. So it transforms the log 2 to a natural number. And that is going to be your size factors of every sample, right? So you calculate a size factor for every sample. And the only thing you do when you have those size factors is divide your counts by that size factor. So what I'm trying to tell you is that doing all those steps of doing log transformations and the geometric means, you're trying to remove extreme outliers of counts that may make your normalization process be biased to very weird numbers that may not be representative of real signal. Again, the median of the factors also helps to just focusing on housekeeping genes. There's another thing, right? All this process where DSeq2 is doing is assuming that most of the genes are not differentially expressed, right? Because you are removing the ones that have 0 counts, so they are not taking into account those, but you know that's not going to be differentially expressed. You're also removing extreme cases that may be also differentially expressed. So it's making the assumption that most of the genes are not going to be differentially expressed. Another thing is that through this process, we've been using all the samples, right? So I told you, you calculate the geometric mean across all your samples. It doesn't matter if it's replicate, if it's condition 1 or condition 2, condition 3. So this has an implication. Do you know what implication is this? When you use all the samples in your data set, right? So the implication is that, let's assume you decide to remove one condition from your analysis next day and focus only, I don't know, you have multiple conditions, right? Wild type, treatment 1, treatment 2, treatment 3, and all the replicas and everything. One day you decide, I want to remove treatment 3 from my analysis. When you remove something from your data set and you do the process again, the scaling factors may change because you are removing one of the replicates, right? So generally what we recommend is always do the analysis with the whole data set. Okay, so whatever you have, the samples you're being collected, do the analysis with the whole data set, even if you have a lot of treatments. So the more samples you use, the more accurate of the normalization process is going to be. If you start removing samples, the size factors may change. I mean, you may need to make that decision later because maybe you have a condition that is weird, extreme, the PCA showing there's a lot of variation in that condition. Then there's other reasons why you want to remove them, right? But the first pass, you should always consider to include everything in the analysis. Don't try to do pairwise comparison where you decide, I'm going to start comparing wild type with treatment 1, and later on I'm going to do wild type with treatment 2, and then wild type with treatment 3, and start splitting your data in that way. DSeq allows you to do the analysis with the whole set at once. Okay, so once you divide the counts by the scaling factors, one of the things that's going to happen, you're going to notice your normalized data is going to have decimal places, right? Probably doesn't make any sense for you in the sense that, okay, now I don't have full counts, but yeah, this is going to happen, right? So you divide by a number, a full count, you're going to get some decimal places in your data. So if at any moment sometimes you get some data from a collaborator and you see that the count metrics have decimal places, it's been normalized. You shouldn't use it with DSeq2. DC2 takes the raw counts, not the normalized counts. Or if you download something from a database, you should download the raw count data and not the normalized count data. Okay, we've seen this. We've gone through the normalization. I'm not going to go through all these details. This is a statistical analysis that I'm not even an expert of. Okay, so what we're doing, right? So here I'm giving an example, right, of what we are trying to estimate. So this is just one gene of our data set. We have a bunch of replicates for what we're going to call, we have some animals that are normal, right? And we have some animals where we sleep deprive them. This is just an example, right? And actually is the example we're going to use today. So basic DSeq2, what it's doing, it calculates the baseline or the interception if it was a linear regression, and then it estimates some sort of change, right? Our fold change. So we're trying to figure out if this difference is actually different from 0, right? If our fold change is significantly different from 0. That's what it's trying to do. And I'm showing this because when we get more complex models, it's for me a list. If I need to deal with a complex model, I like to create my own drawing of how I'm taking the approach, because fold changes gets even more complicated when you're trying to do more complex models. And if we have time, I hope we have time, I'm going to show you a few of those. So I'd like you to have this kind of picture, that DSeq2 calculates the baseline or the intercept in our formula, and then estimates if this difference is significant or not, right? Yes? Questions about this? No? So this is what we're trying to do, estimate the baseline and estimate the coefficient of the condition, so our fold change, and if it's different from 0. And it's quite similar, the approach we do with when we have a continuous variable in the x-axis. We don't have, right? We have a discrete variable in this case, and we estimate the slope. Okay, so I give you an overview of how DSeq2 works, and I think you know the trick here. Oh, sorry, I didn't start the. Oh, sorry. Just give me a second. So if you have questions, please ask them now while we wait. So the question about the whole numbers. So we start with the raw counts, which are whole numbers. But when you normalize them, you're going to get some table that looks like with decimal places. I was trying to show that because sometimes you may try to download something from online and you get a table of the counts. And if you see decimal places, it means it's been normalized already. And you don't want to use that with DSeq2. DSeq2 only takes the raw counts.
Basically, this number is the number of times these copies were expressed, whatever we are getting raw
Yep.
counts.
Yep.
So
Yes.
sometimes it's in million and sometimes it's in very less numbers. So there is lot of variations
Yep.
in the expression of the gene.
Yes. Yep.
So that is telling us this much low expression as compared to the.
You shouldn't compare across genes, right? Because of what we said, the length of the gene is also important in this case. If you have a very short gene, you're going to have less read. If you have a long gene, you have a long read. So generally, a gene expression is not designed to do analysis across genes. That's not the idea. It's across samples or conditions.